Good afternoon, friends, in Hong Kong Genome Institute, as well as through Zoom. My name is Brian Chung. I'm the Chief Medical and Scientific Officer of the Hong Kong Genome Institute. It gives me great pleasure to introduce our inaugural speaker for the Gene Club. So Dr. Jack Yao is our speaker today. Dr. Jack Yao currently serves as the Chief Scientist at Tencent AI for Life Sciences Laboratory. His research interests include computational genomics, medical imaging, precision medicine, and surgical navigation. His papers were cited more than 23,000 times, according to Google Scholar, with an H index of 62. He was listed in the top 2% of scientists worldwide in both career-long achievement and single-year achievement categories. He serves as associate editor for several AI medical journals and area chair for several AI conferences. He holds a PhD degree in computer science from Johns Hopkins University of Baltimore, USA. Dr. Yao is a fellow of the Institute of Electrical and Electronics Engineers and a fellow of the American Institute of Medical and Biological Engineering. Today, the topic of his talk is enhancing the analysis of genomic data with AI. Dr. Yao.
Today, I'm excited to present some work in my group about enhancing the analysis of genomic data with AI. So, as I'm deeply involved in AI for life sciences, and I actually firsthand witness the great potential of AI in improving, in enhancing scientific discovery and improving healthcare. So I think there's a great opportunity in this intersection between AI and genomics. So as we probably all notice that the gene sequencing technology had advanced greatly in the past decades. And I think right now the gene sequencing cost actually dropped dramatically. As we know, actually in 1992, the first generation sequencing cost of the whole genome cost about $300 million. And then when at 2005, when the second generation sequencing is coming out, the cost is reduced to $3 million. And right now, actually, the cost dropped to about $100. So this is like exponentially, and then all actually decrease beyond the Moore's law. And also, actually, right now, the gene sequencing technique, we can actually complete sequencing in just a few days, and the accuracy is above 99.5%. And as a result, now the gene sequencing actually has extensive clinical application in pharmacogenomics, genetic testing, and prenatal screening. And so actually the cost dropped dramatically, and then the data also increased exponentially. They all beyond the Moore's law. And now actually we have a large amount of genome data. But I think for all this data, actually there's an estimate only 20% of these data have been really used and analyzed, because they're still lacking advanced data processing method and algorithms. And in the past 10 years, there's further breakthrough, especially in single cell and spatial sequencing. And before, actually, when we sequence a sample, actually usually we sequencing all the cells together. So and the sequencing result actually is the sum or the average of all the cells. But right now, in the single cell sequencing, we are able to separate each cell, and then we are able to get the DNA sequence and the gene expression, protein expression, and metabolic concentration of every cell. And in this way, we would be able to know each cell, and then the environment of the cell, and then the heterogeneity of all the cells in the tissue. And this actually provide tools for new biological research. So actually, in the past few years, this single cell sequencing and spatial sequencing has named as the method of the year by Nature and Science in a few years, like 2018, 19, 22, and 24. So right now, actually, for each cells, we are able to get all the different properties, and then this actually generate a large amount of data, and then it also bring a lot of challenges for data analysis. For example, and now we have a very large data set for each cell, we have like 20,000 genome sequencing, and then actually for each samples, we probably have thousands of cells. And then also this is a very high dimensional data. And then because right now we have all different platforms and techniques, and then usually all this data has some batch effect, and integrating them also bring a lot of challenges. And also all this data is very compressed. We have for all these genes, they have gene regulation and protein also interact with each other. So this kind of relationship make the data very compressed. And also we have different modality of data like DNA, RNA, protein, and also we have sequence and also structure and also some description. All these modality, if we want to get a whole picture of this cell of gene, we need to integrate all these modality data together. So this make the data analysis even more challenging. And actually, in the past 10 years, the AI technology also advanced rapidly. We have many advanced AI techniques to analyze very compressed data, like self-supervising learning. In this way, actually, we can use this method. We can actually get the inherent data characterization by the data itself. And we don't really need annotations. We just need a large amount of data. We would be able to, using computer algorithm, and you can do some self-supervising training. And we also can build a graph neural network to model the compressed relationship between data. And also the multi-modality learning actually can integrate data from different sources, from different modalities. And all these techniques actually together in the maybe the past 2 or 3 years, there's a new AI technique called large foundation model. They actually built on top of all those advanced techniques. And then this large foundation model actually already bring revolutionary change to the world. And actually, we believe this kind of model is very suitable for genomic data analysis because this data, as we say, is very compressed and very large. And then usually we don't have a lot of annotated data. Then we need some technique to actually find out what the data are really about using this large model. And then in our group, actually, in the past 3 or 4 years, we have been working building AI models to analyze this genomics data, especially those single cell data and then spatial sequencing data. And then one of the first model we built is called scBERT, which we actually start in 2021. And then this model actually, we actually first introduced a large language model into this single cell transcriptomics analysis. And because this actually was before ChatGPT was widely known by people. And then we started this work in 2021, and then our paper actually was published in 2022. And then actually ChatGPT came out maybe 1 month after our paper was published in Nature Machine Intelligence. So this is the framework of our model. So it kind of have a two phase. The first phase is the pre-training, the self-supervised pre-training, and then the second phase is the supervised fine-tuning. So this framework actually now is very popular and well known. But at the time when we started work, it's still it's kind of a normal idea, and then it's especially for this gene analysis. And then so this one actually we in the self-supervising pre-training we use the mask language modeling technique. So we actually mask some of the gene expression and then ask the computer to reconstruct the data. In this way, we would be able to, if we do it many times using a large amount of data, we would be able to train encoder and embedding that actually would be able to characterize all the data, and then actually we would be able to reduce the dimension of the data. And then in the supervised fine-tuning, we actually need a very small amount of annotated data. We would be able to train high performing models that actually can actually have a very high performing and then in those tasks. And actually this kind of model usually have better generalizability than the previous model actually need a lot of annotated data. So actually, we do some performance evaluation for our model, and then we test our model in 9 independent data sets with 8 state-of-the-art method. And we actually noticed that our method has much better robustness and generalizabilities, and has higher performance than all classic method. And our method can also have potential for a wide range of application in various scenarios, such as cell-specific embedding and gene-gene interaction and batch collection. And then we can also use this model to discover normal cell type and then normal marker genes. And then so because this is kind of the first work, this kind of model in the gene analysis. So actually our work received great attention in the past 2 or 3 years, and it has already been cited more than 300 times and downloaded 50K times. And because I think this technique, I think is kind of the first of this kind, and then the Nature editors noticed that, and then later actually published news and views about this technique, and they think this large language model can actually be a powerful tools for gene analysis. And then later they also organized a third-party team to test this method. So and then they actually published a usability report using this model on a totally new data set. And then they also noticed that actually the performance of this model actually is much better than the existing method. So that means this kind of technique can really be used in a new data set and then have better usabilities. And the second job we were working on is for single cell proteomics. And we all know that actually protein actually is really the molecule actually regulate the cell activities, and then it's more direct. If we know the protein concentration, we would be better reflect the cell state. So actually, proteomics actually is currently is a frontier of research area. So people try to get better, more accurate protein measurement within a cell. And right now, actually, there are 2 kinds of technique for single cell proteomics. One is called antibody based method. And then this method actually they bind the antibody to the epitope of the protein, and then on the other protein, and then the barcode could be measured and then used to quantify the corresponding protein. So this method actually can be very accurate and specific to certain protein. But the problem is that it's very low throughput. Well, we can only measure up to 100 protein per experiment. And then the second type of technique called mass spectrometry based method. And then in this method, the protein are digested to peptides. And then each peptide are barcoded and analyzed. And then actually they are measured. And then later they will accumulate all these peptides to measure the concentration of proteins. So this technique actually can be very high throughput. We can measure up to 10,000 proteins in one experiment. But the disadvantage is that because this method actually right now is not very accurate and has some noise and batch effect, and also the peptide measurement is kind of uncertain because some peptide maybe have very similar mass, because this is based on the mass of the molecule. So there's some uncertainty there. So in order to make this technique work well, we really need to have some very good data analysis and processing technique. But this is right now is a frontier of proteomics, and there's still a lot of active research in this area. And here is actually the pipeline how currently these mass spectrometry data are actually processed. It actually has 3 stages. So PSM stage, peptide level stage, and protein stage. And so in the first stage is the PSM is peptide spectrum matching. So here we actually need to match each peptide to a spectrum, and then because there's the noise I mentioned, it needs to have some quality control steps, such as filtering and data normalization. And so actually we need in this step we need to get rid of many of those low quality peptides, and then all the PSM, and these PSM now later aggregate into peptide, and then for all these peptides we also need a series of quality control steps like normalization and filtering, and then later after that this peptide will be aggregate into protein, and again we also need normalization, imputation, and batch collection, all these quality control steps. So, and after that, then we can do this downstream analysis. So that means that means actually there's so many steps to do all this work, and then all these need to some of the normalization and the imputation collection all need computer algorithm to do it. And traditionally, in the traditional method, actually usually use like k-mean clustering and this kind of traditional technique, but usually it doesn't work very well in some cases. So my group actually try to see whether we can use this more advanced AI technique, like the technique I just mentioned, like self-supervising learning, constructive learning, and graph neural network to actually analyze these single cell proteomic data, make them more unified, and so that we can integrate data from different experiments, and so that we can do the analysis and comparing them. So we actually proposed a model called scPROTEIN, which is a unified protein embedding for single cell proteomics. And actually, our model has 3 stages. And then the first stage we call peptide uncertainty estimation. And in this step, actually, what we do first, we actually get all the low peptide data and then put them into a matrix using a one-hot encoding. And then this is like a 20 by n matrix. The n is the length of the peptide, and then 20 is like the 20 normal amino acid. And then we have a few CNN layer to actually process and then this matrix, and then later we flatten them into a one-dimensional vector, and then we actually use a linear regression model to estimate the expression of the peptide, and then we compare this estimate peptide expression with the observed peptide expression as a loss function, and then we can actually using a multitask heteroscedastic model to estimate the uncertainty of the peptide and predict peptide. And then later we actually use this uncertainty as a weight to build a weight matrix. And then this weight matrix later can be used to adjust the measured peptide, and then we can get the uncertainty guided aggregation to normalize get a normalized protein data. And later we can use this protein data to do the further analysis. And then in the second stage, we also further process this data to reduce the batch effect and then the data missing problem. So here we use self-supervised learning technique. And then here actually in the stage 2, we have a step. So the first step is we use graph modeling to actually build a graph for this data. So in this graph, every node is a cell, and then the property of the node is their protein expression, and then the edges between the cells are the actually correlation between of these two cells, and then using their protein profile. And then we actually use a technique called contrastive learning. So, and what this technique is about is actually we perturb this network. So actually, this is kind of data augmentation method. We make small change to this network, and then you will have different configuration. But because this change is very small, so they should still represent the same tissue. And then we use the contrast between the different configuration of this network and to actually guide the training of the model. And actually, if we do it many times, at the end we will be able to get the graph encoding of this network. And then so this would be able to get a better representation. Actually, we will be able to handle the data missing problem because we have a graph, and then if we have missing data there, and then its neighbor would be able to fill in the data. And the second step is mostly for denoising, and then we also again we're using some contrastive learning based method, but here we instead of using the entire graph, we only use a subgraph, a small subset of the graph, because the noise usually is local. And then within each subgraph, we also doing some the similar contrastive learning, and also we also so doing this called attribute denoising step. And then the topology denoising usually is kind of we actually doing some perturbation between a subgraph by adding or removing edges between a subgraph, and then also we use that as a contrastive learning scheme to learn the configuration topology between all these subgraphs. So after doing all these ways, we would be able to make the denoising of this graph, and then we also will solve the data missing needs problem. So after that, after we train that, we will be able to get a trained graph encoder, and then given a new data set, new sequencing data, we will be able to fit into this encoder, and then we get a learned new embedding, and then this new embedding will be consistent through all different batches. So we can, in this way, we will be able to perform different downstream tasks like cell clustering, batch collections, and then clinical analysis, also spatial protein analysis. So these are the data we actually used for training and test our model. So we actually using data from different biotechnology like the scope, N2, Plastier, and all these are all different techniques, because right now we have so many different kind of proteomic techniques, and then it's still not unified. And then we hope that the computer model can help unify some of these techniques. And also these we have different number of cells range from hundreds to tens of thousands. And also the protein, we have like some data we only have like 40 protein, and some has more than a few thousand proteins. And all these cells have different cell types. And then we use this data to train and test our method. And here are some evaluation result. We can see, we compare first task we compare is for cell clustering. So we can see using our technique, we can better cluster cells of the same type. So we compare it using some of the state-of-the-art method, and our method has a bad performance. And here is a batch. The second test is the batch collection one. And when we see that on the left, we can see actually the data is pretty. That's before using the original raw protein expression data. We can see it's quite noisy, and then all the different cell type actually you don't see the uniform properties. But when you look at the one on the right, our scPROTEIN embedding, we can see very consistent and uniform profile of the same cell type, even they are from they are acquired using different technique. Actually, here we have two techniques. One is nanopod, and the other is N2. But originally, the low data they are quite different. But the scPROTEIN embedding, we can see very good embedding. And also the data integration one, we can see actually the same cell type, if we're using the low data, they are not clustering together. But after we're using the scPROTEIN embedding, they are clustered together. And also we can use this embedding to find the marker protein from clinical proteomics data set. And also this in this spatial proteomic data set, we can see actually using a raw data, we cannot see a very good the boundary and region of different tissue type. But after the embedding and also the denoising, we can see actually they have very good spatial distribution. And actually, this method was published in Nature Method last year. And the next job is actually about we try to find a relationship between RNA transcriptomics and proteomics. And we all know about the central dogma, right? The DNA transcribe, this actually describes the genetic information flow in biological system. And then the DNA transcribe to RNA, and RNA translate into DNA. So this actually is the basic law for biological system. And then if if the translation between RNA and protein is like a one-to-one mapping, then it would be as easy as translating from one language to another language. But actually, the translation from RNA to protein is very complex. It's not a one-to-one mapping, and it usually involves many RNAs, and the RNA regulation network was involved into protein, and then different protein has protein-to-protein interaction. So this kind of interaction also will affect the protein expression. So if we actually if we plot the RNA and the protein expression data, you will usually like the plot here. We cannot see really a linear relationship. It's a much more complex relationship, but there must be some relationship there. So we try to see whether we can develop advanced computer model, especially some of the new large foundation model. Can we use this kind of model to model the relationship between RNA and protein? This model actually has encoder and decoder architecture. So so both these encoder and decoder are transformer based, because we know a transformer actually is the basic module for the ChatGPT and all these large foundation model. And then in this encoder, the input is the gene ID and the RNA expression data. And then we actually for this gene ID, we have a model to map them into 80,000 mapping, and then we have using a linear embedding to embed this gene expression model, and then in the encoder transformer, we actually use a new favor plus mechanism for the self attention, and then to address the long sequencing, and then the output of this encoder would be the encoded gene expression. And then in the decoder part, the input is the protein ID. We're using the same position encoding, and then the output of the encoder, and put them into another transformer-based model, and train a model to actually output the protein abundance value. So after doing this encoder-decoder base, we will be able to translate the RNA expression into a protein abundance. And then actually, the model training, we actually do it in two stages. And then in the first stage, because this is a single cell translator, but actually right now it's because we need a paired RNA and protein data. But right now, there's not that many paired data for single cells, but there's quite some bulk data. So actually, in order to fully make use of all the existing data, we actually make the training into two stages. And the first stage, we actually pre-training this model on paired bulk data, and then the second stage, we pre-train it on paired single cell data. So this one actually, all this data will go through this bulk encoder and bulk decoder, and then this model will be transferred into the second stage and using the single cell data to do the training. And then here is the data we use for the training. All of them are actually public available data. We have over millions of these data. And then this model actually currently is about 100 million parameters. And then here are some evaluation of this model. we can see the plot of the measured protein and then the predicted one. And this is an example for the protein CD45. We can see actually they have a pretty good correlation. That means the predicted value is match very well with the measured the protein value. And then actually not all the protein can be very accurately predicted. And then we actually randomly select 80 of proteins and then rank the predictability. That actually is how good the prediction is. And we can see actually some of the protein we can actually make it like over 90% accurate, and some of them maybe only 40% or 30%. And then we also compare our method with like five independent data set with 7 existing methods. And then our method actually have much superior performance than all existing methods. And we also compare our method without the first stage, only use a single cell data. We call scTranslator scratch. And we can see actually our 2-stage training scheme actually make the performance much better. And here are some downstream application comparison. We can see using the predict protein, we can actually get much better clustering of the same cell type. And here is actually we try to cluster all these immune cells, B cell and different T cell and then NK cell. And we can see actually the first one, the first one is the predicted one. Actually, we have much better clustering. And then the second one using observed protein, we see all these the batch effect. And the RNA actually we are not able to very good separate cell from different type. And this is a measure of the performance metrics. And we can see actually the predict protein actually have much better performance. And we also actually doing some virtual we actually virtually knock out some of the gene and then see how it will affect the prediction. And we can see actually by knocking out some of the gene, actually the predict protein also actually abandon also job. And using the attention mechanism of our model, we actually not only can actually predict the protein abundance, we are also able to actually discover the gene regulation network using the attention scheme mechanism in the encoder. And we also can discover some of the protein and protein interaction using the protein network. So actually, this model we not only can actually do this translation, we can also using this model to make some new discovery. And this work was recently actually accepted to Nature Biomedical Engineering. And also actually, as I say, actually, spatial sequencing actually also a very active research field, and then actually have very great advance in the past few years, and then it also named as a method of the year in the past few years. And then in this technique, we are not only can actually get the sequencing of the cell, we can actually get their spatial, we can preserve their spatial location. So in this way, we would be able to know the microenvironment of this cell, and then which and the cell interactions, so that we can do a lot of biological research using this spatial resolve omics technique, such as tissue homeostatic. We can build the relationship atlas. This is especially useful for those neuroscience. They want to know how neurons interact with each other, and then using this technique, they will be able to get the property of each neuron and then their interactions. And also in cancer biology, we can use this technique to see how this cancer progress and how it start and then progress and then how it will be react to different treatment and also different disease development. So this is kind of a very important technique in biology. And but so also because we have this new data set, new kind of data, and because of this special omics data, because they need to preserve the cell location, so it's not very easy to multiply the cells. So in this kind of data, the data noise is usually more severe than the regular single cell data, and then also have worse data missing problem. And so actually analyzing this data is also more difficult. And the other thing is that this data usually is very huge. One, because it also has an image, and one image usually have like 10,000 by 10,000 pixels. And how to efficiently analyze this data is also a very important thing. And so we actually developed a model called Spatial-ID. So for this cell type annotation for spatial transcriptomics data. And this model also has 3 stages. So in the first stage, we actually use get a reference data, reference single cell data for the same tissue type. And then we build a deep neural network using this single cell data. And this model could be the model we just talked about, the scBERT. And this pre-trained model we can use to get this spatial data there. We can get the probability distribution of this data, and then later we can transfer this distribution into our process data, and then guide the cell type classification. And then in the second stage, actually we actually also have trained 2 encoder. One is called autoencoder to encode the gene expression data, and then the other called spatial variational graph encoder, actually to encode the spatial relationship among all these cells. And then this is also this is using a self-supervising learning, so we don't really need any annotated data. And then and then at the end we can, by guided using the distribution model from the first stage, we will be able to annotate the spatial data very accurately. And then in the third stage, we will be able to use this model to analyze new data set. And this method was actually published in Nature Communication. And here actually is some performance evaluation of this model. And we compare this model within 7 existing methods on 5 different data sets. And our model actually have the best performance. And also, this model is not only very accurate, it's very fast, because we are using this deep learning model. And then once it's trained, it's very efficient to employ this model. So the speed of this model is kind of more than 10s or 100s times faster than some of the traditional methods. And then we also can use this to analyze this 3D data set. And also we do some simulation experiment to evaluate how this model perform when there's some dropout, when some of the data are missing. And then we can see actually even the data, some of the data missing like 50%, we can still get a relative good performance. So actually, using this model, we collaborate with Chinese Academy of Science, Academy of Neuroscience, to build the cell type atlas for macaque brain, because macaque brain is like 1/10 of the human brain. It actually has like 80 billion neuron cells. So it's kind of very large data set. And then actually, it has many different neural types. And then so using our model, we actually built the first single cell resolution cell type atlas and for this pre-made brain. And then we actually be able to classify all the neurons into 254 different cell types in over 143 cortical region. And then, yeah, so this work actually was published in Cell in 2023. And then here we actually built the entire pipeline to analyze data because this data is so big, and then no existing method can actually analyze it, but our method will be able to process like hundreds of slices, maybe just within a couple of days. So we can actually process all this data. And actually using this model, actually we actually collaborate with different research group in China on different like brain atlas project, like the cortical cerebral cortex and mouse brain, and also like hippocampus, and then all these actually result in like publication in Cells, Neurons, and Science in the past 2 years. And then we also have a few ongoing paper actually still under review. So this is kind of like AI for science. So actually AI kind of example of how AI can actually help doing scientific discovery. So it's so-called AI for science. And I think in the future probably will be more and more this kind of work that actually when you do scientific, you have to probably have some AI involved to help you accelerate this research. And we also doing some because we also know the data is very important for scientific research, and also very important for AI. So we actually built the largest spatial omics data set by far, and then this data has over 500 million cells, and we not only built the data set, we also have a set of processing tools that can be used to analyze this spatial data. And then we have a website actually be public open, and then actually right now this app has over 10,000 access every year. So I think it actually efficiently actually help this research field. And then this database was also published in Nature Method. And we also collect a single cell proteomics database, with like like 300 about 400,000 cells. And then this is for single cell proteomic data. And then we also have this a website and also a processing pipeline to analyze this data. And this also published in NAR last year. And the next I'm actually going to talk about some foundation model we are building for genomic analysis. And then we actually built a CD GPT. So this is a GPT model for genomic language. And then this one actually we are actually using all different type of molecules in a cell, like DNA, RNA, and protein. We actually integrate them into one single big model. So like most current model, they only use one type of data, like DNA or protein, but we use all of them. And mostly the data set is public data set, like the UniRef 50 and sequence left sequence data, and we use like I think billions of data to train this model. And then also so this is a multimodality model, and then we also train this model in multiple stage. So in the first stage, we train a single modality model. So we actually are using each single modality data separately to train it to train this model in the first stage. In the second stage, we actually pair up the data through those central dogma, like if the RNA related to protein in the translation, we actually pair them up and then concatenate the data and then send it to the model for training. So this kind of a paired modeling. And then in the third stage, we actually also using the structure, like the protein structure. We also integrate this information in the model. So that this is a kind of a real multimodality data. It has all these different properties of these objects. And then this model actually is kind of also based on transformer, but it is an decoder-based model. We don't have any encoder, so we actually directly send this sequence into this model, and then we use a BP tokenization method to encode all these different sequence, and then we also use low precision embedding, and then also this model. The other thing actually this model can do is it can perform different kind of task. So we have different output headers for different type of tasks. So for example, for a generation task, like if we want to generate a new protein or a new gene, we can use the linear layer to predict the next token. It's just like right now most of the ChatGPT like predict the next word. We can predict the next residual of the sequence, so that we can generate new protein, a new sequence. So for this generation task, we can do using this head. But for the sequence prediction task, like we want to predict the attribute of a protein of a DNA, we actually have a new set of head using a set of dense layer to train a classifier to predict the property. And then for the token prediction task, like we can actually predict the property of each residue or each token. And then again, actually the input of this one is each token. And then also each token has a classifier. We can do a multi-classifier head. And then we also can do a residual paired prediction. That we can predict the relationship between two sequences. And then again, you build a confusion matrix, and then from this matrix, we have a few layers, and then we can actually predict the relationship between different pairs. So this model is kind of not multimodality model has a versatile function. You can perform essentially all kind of genomic analysis tasks. And actually, we actually have compare our model with some state-of-the-art on a few downstream tasks. All these tasks have different type, like solubility prediction, supply side prediction, and also like the RNA and protein interaction prediction. And there's some benchmark data set that actually you can evaluate the model performance. And then our model currently actually have better performance than some of the shorter methods. And we can also use our model to generate new proteins. And we can see actually the protein generated by our model actually had pretty good PLDDT. That actually is a measure of how this generated the structure of the generated protein similar to the nature proteins. And then if it's good, then mean that this protein actually can be developed, and then you actually can actually have certain functions. And also we can look at the structure of the generated protein and compare with the nature protein, and we can see they actually have similar structure. That means they could have the similar function, because we our model to generate this kind of protein, and then it actually generates some protein similar to what we asked it to design. Okay, so actually, so right now actually we talk about we have all these gene data, and then now the data has a large amount. But in order to actually doing population research or doing some systematic biological research, we actually need a large amount of this data, and then all these data actually need to be data acquisition need to be well designed, and then it follows some similar protocols, and then that actually is like people realize the importance of this actually put all a large amount of data together and then building something called a gene bank, and then actually I think right now many countries actually put a lot of importance in this building this gene bank, and here is actually the list of 15 biobank is currently largest biobank currently in the world. You can see most of them actually is in the United States. They have 7 of them, and then the UK have 4 of them, and then there's a few others around the world. Like in China, they have the GKB that is about chronic disease, and then there's also other smaller one or not public gene bank around the world, but actually there will be more and more of them, because I think right now the United States actually they already start to restrain the access of the gene bank or their biological data, especially to countries like the competitive, they think like competitor like China. So I think more countries probably in the future will start to build more biobanks. And I think the UK biobank actually currently is one of the most famous one, probably one of one of the largest one in the world, because this project actually started in 2006 and currently contain about 500K individuals gene data, and then they also follow this up for more than 10 years. And here are actually the gene bank data usually have. It has the basic information about the individuals, and then have their genomic data and also imaging data and health-related outcome data and biomarkers and activity data. And using all this information, we can actually get a comprehensive understanding of these individuals. And then currently there's many research have been conducted over this biobank, such as doing disease prediction and diagnosis, and then for disease risk factor analysis on drug target assessment. And there's many research and publication on this one, but most of them actually currently are based on classic method like statistical analysis. And currently, because this data set usually is very big and very compressed, and then multi-modality and heterogeneous. But currently, I think there's still no published like foundation model building on this kind of gene bank data. And then we are actually currently start to work on building foundation model, multi-modality foundation model on gene bank data. So this kind of has actually 2 models. And then the first one is like we build it for genomic foundation model, because genomic data is kind of very different from other type of data. And then this model we can use like based on the CD-GPT model I just talked about. You will analyze different kind of genomic data and different kind of protein data. And then you actually can, the input would be like the gene data and then the mutation data, and then output would be some embedded information. And then for all other information, like the patient basic information or imaging or diagnosis data, we can build a multi-modality clinical foundation model. And this model we actually can use, like we can convert all this information into structured text, and then using a DeepSeek model, and then fine-tune the DeepSeek model to understand all this information, and then refine the model to build a foundation model. And then at the end, we can actually fuse these two models using some another fusion modules, and then we will be able to come up with a model that actually can analyze this compressed multimodality data. And to build this model, actually, we can do it in 3 stages. First stage, we probably doing this basic foundation model construction. We actually can use a larger amount of like public available data, like this 1,000 genome data or this UK Biobank data to build the initial foundation model. In the second stage, we actually can specifically fine-tune this model for a specific gene bank or a specific large study clinical trial or cohort. And then we can actually freeze some of the model parameters and then fine-tune some of them using these specific data. And then the third stage, we can actually deploy it deploy this model to analyze the individual data and cohort study. So this model actually data can be actually deployed into a cloud. We can use some cloud service like storage, security, computing, and privacy to use this model to do a large clinical trial or cohort study or individual disease analysis. So this is kind of an ongoing project. Actually, maybe we are hoping can get some collaboration from people that are interested in building these things. Because right now we are currently still in the first stage, building the foundation model using the public data. So that's actually some of the project we are working in the past 3 or 4 years. And then we already know witness that actually there's a rapid advance in both sequencing technique and the AI method. And then I think our clinicians and scientists to really collaborate this exciting intersection of AI and genomics to move this field forward. And we actually open source some of our method models in GitHub. So if you are interested, you are welcome to try it. And there's some other work I didn't mention here that we also list there. Okay.
Thank you very much, Jack. This is a great lecture. I hope that all of you would agree with me that the work that you're actually doing is really cutting edge. So if you allow me to summarize, you can see a pattern. The pattern is whenever there is a new method published in Nature Method in the same year or the next year, you will have an AI solution to look at the data. And the data not only allowed it to be analyzed in a more rapid, efficient way, there's less noise, is more efficient, and you have more downstream applications. So I think this is really the trend. And he always benchmark his work with other platforms, and always there is evidence of superiority. So this is really a great lecture, and it shows us a lot of possibilities to use AI, not only in healthcare, in particular in genomic medicine. I have already collected some questions. In the interest of time, I think I'm going to those more philosophical ones to ask you. When we always talk about AI, a lot of us here are happy and worried at the same time. When do you think that AI is going to replace our scientists and our clinicians?
Yeah, this is really a theological question. Yeah, I think, some people say AI probably will replace human in the future, but I think probably not in the recent years. But I think right now actually more AI is more in the assistant role, right? We all say AI for science, AI for industry, AI for many things. So I think AI, I think we probably there's a possibility AI probably will replace human in the future, and then I think there's some ethical issue there, and then we probably, but we at the current stage we probably shouldn't too worried about that. I think currently the AI is still not there yet. I think, and even for some of the work I'm talking about here, we can see there's still a lot of room we can improve. There's still a lot of work. Like we need to develop new AI models to analyze the data. So still, I think AI still is in a very early stage. I think they don't really have real intelligence yet, but I think they can really make our work more efficient. And I think they can very good at analyze those large data set, very compressed data that actually human probably not very good at analyzing.
There was a saying that AI can do tasks better than human, but in order to perform a job, you still need a human. So a job is really the integrations of everything. So the second question, I think, is probably coming from a younger person, a trainee. They were asking if they are going to study more. In the field of AI, for example, what would be the interesting area that they should embark on in their future studies?
Okay. Yeah, I think AI could actually apply in many different ways, right? Like in medicine, in biology, and also in many other things. Actually, we are already in the production and many ways. And I think if you are good at in any area, you probably can thrive, right? So I think right now you probably need to build a very good base. You need to although we already have a lot of open source resource there you can use like all these models, but you still the underlying still is the mathematics, the physics, all these things, and then maybe the computer programming. So for younger people, you still at the beginning you need to build a very good foundation, and then how to apply this AI to which area. And I think that probably is up to your interest. Because if you are interested in medicine, you probably can try to see how you can apply AI in medicine. But if you're interested in like material, you can doing other things. And I think the interest probably is more important.
I think it's really the limitation is really the sky, right? I think that this is very true. I'm totally aware of the time. I think, Jack, you're going to stay for a while to answer more questions. But at this time, I would like to invite you to take a seat first. I would like to do some promotion work for our next Gene Club. This is great opportunity for us to learn from each other, and the next scientists, clinicians, and entrepreneur is actually Dr. Haiming Lai, all right? So you see that this is a news covering the company that he has founded, Illumos, right? And he is actually from the Department of Chemical Pathology of the Chinese University of Hong Kong. So he is a scientist, he is a clinician, and he is an entrepreneur. As a clinician, he actually sees the problems in the clinical area. He sees what are the potential area that needs improvement in his patients. As a scientist, he uses innovations to discover new methodologies, new technologies. And as an entrepreneur, he actually convert his scientific ideas and discoveries into commercializations so that through the process, he can help more people. So you see that this is the promotion for the next Gene Club is going to be on June 24th. And the topic Dr. Lai is going to share with us is spatially resolved molecular pathology in 3D for precision phenotyping. And it has great opportunity and applications in the field of cancer diagnostics.